While you're doing this, maybe I can introduce you, Sophia.
Yeah,
So everyone, welcome. Thanks for being here. This is a session organized by Florence Nightingale Day, Statistics and Data Science. I'm really honored to chair this session, and a big thank you also to Shili and Molly for organizing this session. So today we'll hear three outstanding students who apply statistics and data science to real-world problems. And we are actually going to start with Sophia. Sophia, are you going to be able to share it or no?
I think there's an issue with my system settings and trying to share PowerPoint.
Okay.
So that's the only issue is I'm having trouble pulling up the PowerPoint.
I can share the one that you send me if you like.
Okay, that would be great. Thank you.
Okay, yeah, sure. So let me first open it. Can you see it now?
Yeah.
Okay, and you can just give me a clue when you're ready to.
Okay. I'll give you, does a thumbs up work?
Sure.
Okay. Thank you. All right. Hi, my name is Sophia Selassie. I'm a senior at Athens High School, and my project is titled Investigating the Effects of Two Emotional Regulation Strategies on Runner Performance. So I'm a high school distance runner. I run track and cross country for my school. And one thing I've learned about the sport of running is that there's a lot of pressure that comes around competing. A lot of athletes feel anxiety towards races and like these pre-race nerves. And so as an athlete trying to reach the next level, I often ask myself, well, what can I do to enhance my performance? And the first thing that comes to my mind is how can I better manage my emotions before a race? So last year I did a science fair project that looked at the relationship between a runner's personality type and their use of different emotional regulation strategies. And so this past year I kind of wanted to examine the effect of different emotional regulation strategies on a runner's performance. Go to the next slide, please. Okay, so what is emotional regulation? Emotional regulation is the process by which a person consciously or unconsciously evokes, reduces, or intensifies a certain feeling state to achieve a desired outcome. So emotional regulation is an important psychological skill for everybody, but it's especially important for athletes who are trying to achieve an elite level of performance. So there's a lot of research that has examined the effects of emotional regulation on performance, but there isn't a lot of research that differentiates between the effects of different emotional regulation strategies and how they impact an athlete's performance. You can go to the next slide, please.
Sophia, sorry. Also, can you change the display settings? So, yeah, swap yes, that one, I guess. Yes, thank you.
Yeah. So, oh, you can go back one slide, please. Perfect. So different emotional regulation strategies serve a unique function to achieve a desired target. So some emotional regulation strategies aim to enhance an athlete's excitement, while some strategies may aim to reduce feelings of stress or anxiety. So runners commonly employ cognitive reappraisal and visual imagery in cognitive reappraisal is the process of modifying your perception of a given situation to change its emotional impact. So for a runner, this could mean viewing your race as a challenge instead of viewing your race as a threat. Visual imagery involves imagining a positive outcome of your competition to respond to the demands of the event. So therefore, the purpose of this science fair project is to examine the effects of cognitive reappraisal and visual imagery on a runner's performance, which leads me to my first research question. Is there a difference in a runner's performance time based on the type of emotional regulation strategy used, cognitive reappraisal or visual imagery? My null hypothesis is that there's no difference in a runner's performance based on the type of emotional regulation strategy used, and my alternative is that there will be a difference in a runner's time performance based on the type of emotional regulation strategy that runner uses prior to running. Next slide, please. So my second research question, is there a difference in a runner's rate of perceived exertion, RPE, based on the type of emotional regulation strategy used? My null hypothesis is that there will be no difference in a runner's time. in a runner performance, there will be no difference in a runner's rate of perceived exertion based on the type of emotional regulation strategy used. And my alternative is that there will be a difference in a runner's RPE based on the type of emotional regulation strategy that runner employs prior to running. Next slide, please. My third research question, is there a difference in a runner's heart rate based on the type of emotional regulation strategy used? My null hypothesis is that there will be no difference in a runner's heart rate based on the type of emotional regulation strategy used. And my alternative is that there will be a difference in a runner's heart rate based on the type of emotional regulation strategy that runner uses. Next slide, please. So in order to complete this experiment, I use the rate of perceived exertion scale, the sport success scale, the sport orientation questionnaire, polar heartbeat monitors, Run Scribe shoe pods, SPSS for data analysis, and Sign Up Genius online survey platform. Next slide, please. This project utilized a quasi-experimental research design where a strategy type was a within subjects variable. That is, I had 2 time trial sessions where I manipulated the type of emotional regulation strategy that runner used prior to performance. So data collection involved 2K time trial sessions, which were approximately 4 days apart, and each participant would complete a 6-minute warm-up jog around the track, followed by a series of brief dynamic stretches, and then they would listen to an audio using Champion's Mind, which is an online mental training application designed for athletes. And the audio would facilitate a participant's use of either visual imagery or cognitive reappraisal. And these audios lasted approximately 4 minutes each. So, and I also counterbalance these audios. So half of my participants would listen to the visual imagery audio first, and half would listen to the cognitive reappraisal audio first, just to ensure that the order of these audios isn't impacting a runner's performance, and that it's purely the mental training strategy that they're using that is impacting how they perform in their time trial. So while the participants were running their 2K time trial, they were wearing heartbeat monitors and shoe pods, and this just allowed me to collect additional biometric data around their performance. And I did have them complete their time trials in an indoor track facility just to mitigate like weather variables, wind, temperature, and try to keep that consistent across all my participants. So after they completed their time trial, they completed a brief questionnaire which contained the 3 established scales that I mentioned before, the sport orientation questionnaire, the sport success scale, and then the RPE scale. So as I mentioned, the survey included those 3 established scales, the rate of perceived exertion scale, which was only 1 item, and it measured a participant's level of perceived physical exertion during their time trial. This scale is on a 14-point Likert scale with responses ranging from 6, no exertion, to 20, maximal exertion. The sport success scale, which included 10 items and measured a participant's sport achievement motivation, and I included 3 subscales from the sport success scale, including flow state, attention, and sensitive to error. And this scale was on a 6-point Likert scale with responses ranging from 1 to 6. My final scale was a sport orientation questionnaire, which included 9 items and also measured a participant's sport achievement motivation, and I included 2 subscales from the scale, competitiveness and desire to win. And this was also on a 6-point Likert scale with responses ranging from 1 to 6. Next slide, please. So taking a look at my participant profile, just some general demographics about my population, I had a total of 15 participants complete the 2 time trials, 7 male, 8 female. The average age was 25.4, 46.6% of participants were in high school, and 60% of participants were current students. Next slide, please. These are some more runner-specific demographic information. The average number of years running across my sample size was 13.2. The average number of days run per week, 4.2. 46.6% of participants ran 1 to 5 races in the last 2 years. 40% of participants ran 21 to 30 miles per week. 73.3% of participants identified as competitive runners, and 40% were motivated to run by fitness. Next, please. So before doing any hypothesis testing, I first ran reliability estimates on the 2 scales using Chromebooks Alpha, and so a score above 0.6 shows that the scale is considered reliable. So as you can see here, all my subscales were found to be reliable except sensitive to error, so I did not include data for sensitive to error in my follow-up analyses. And so when we try to examine why this might have come up, I did modify some of the items in my subscale to fit within the context of running the time trial race. And so because I modified some of those items, it may have changed the intention of what the question was gearing at, and therefore would change the reliability of that item and therefore the subscale. Next slide, please. So these are the descriptive statistics for running performance, biometric data, and sport orientation. So a couple factors that stand out here: runners had a faster overall pace while utilizing the cognitive reappraisal emotional strategy. So we can see that their overall average mile pace was 6:32 minute mile while utilizing cognitive reappraisal, compared to 6:48 minute mile while utilizing the visual imagery strategy. Runners also had a faster overall time trial time while utilizing cognitive reappraisal, with that average time for cognitive reappraisal being 8:07 and the average time while utilizing visual imagery 8:17. Runners also had a lower heart rate while listening to the visual imagery session, with that heart rate being 40.34 as compared to 44.89 while utilizing cognitive reappraisal. Other than that, while looking at this graph, emotional regulation had no obvious effect on the other variables. Next slide, please. So in order to address my research question, I ran a series of paired samples t-tests to explore the differences in runners' performance while utilizing cognitive reappraisal versus visual imagery. So I did find statistically significant differences in runners' time performance based on the type of emotional regulation strategy used, and Cohen's d indicated that emotional regulation had a moderate effect on a runner's performance. You can also see here that pace is approaching significant. The p-value has to be below 0.05 and 0.04, so very, very close. So perhaps if I were to get a larger sample size, we may see that mile pace is significant. And so that was also kind of what stood out to me in this project. Is I only had 15 participants and they did complete 2 time trial sessions, but to find statistically significant differences in a runner's time performance while utilizing 2 different strategies was really exciting, and it made me excited to continue this research and see if I could find more statistical significance if I were to increase my sample size and continue this research. Otherwise, there are no differences in biometric data based on the type of emotional regulation strategy that the runner employed prior to running. So this kind of indicates that biometric data may be a more stable variable and not as easily influenced by the intervention. So perhaps biometric data is more a byproduct of a runner's training and their experience in the sport, and that isn't going to be manipulated by the intervention. Next slide, please. So all in all, the purpose of this project was to examine the effects of cognitive reappraisal and visual imagery on a runner's performance in a 2K time trial. I did find statistically significant differences in a runner's time performance based on the type of emotional regulation strategy that runner used, but there were also many other variables that were not found to be statistically significant. So this also leads me to think about the dosage and intensity of the intervention that I'm using. So for one, runners only listen to this intervention once prior to running. And so maybe if I had a more long-term module and I had runners complete, a series of interventions prior to their time trial, maybe they completed a module, like a mental training module geared at either cognitive reappraisal or visual imagery, we may see more of an impact on how that runner performs in their 2K time trial. Then also the intensity of the audios. I think I said before that each audio was approximately 4 minutes long. So maybe with a longer intervention, we may see more of an impact on performance, which would be interesting to explore in follow-up analysis. Next slide, please. So future research could also consider adding a control group, to determine whether implementing cognitive reappraisal or visual imagery into a runner's training is even more beneficial than having that runner, employed no emotional regulation strategy at all. So it just kind of examines the benefit of even incorporating emotional regulation into a runner's training regimen. Future research may also find benefit to exploring a runner's preferences for different emotional regulation strategies. For example, if a runner favors cognitive reappraisal, will that runner perform better while utilizing that strategy? In conclusion, emotion plays a large role in sport performance, and successful runners will know how to manage their emotions to enhance their performance. Thank you.
Thanks,
Oh, you are
Sophia.
muted.
Yes, I see now. I couldn't see my screen in a minute. I mean, because I shared my screen. Any questions for Sophia? Yeah, that was an awesome presentation. Any questions? Okay. All right. Then I think we'll move to Mackenzie, and I'll let her to introduce herself. Do you want to try sharing your screen, Mackenzie? I'm not sure.
Yes.
If not, I'll try to do it again, but it doesn't let me share the PowerPoint.
Mm, Okay, can you see it?
Yes.
Okay. All right, so hi, my name is Mackenzie McCoy, and I presented this science fair project last year as a senior at Northwestern High School in West Salem, Ohio, and currently I'm a first-year nursing major at Ohio State. So the title of my project was Munchin Mice, the Effects of Artificial Sweetener and the Weight Gain of Mice. So some background, today 81% of Americans are obese, making obesity one of the most prevalent health issues facing our nation. As a result, companies have turned to incorporating artificial sweeteners into our food and beverages with the marketing promise of weight loss and a healthier alternative. This is because artificial sweeteners are not metabolically processed by the body, making them 0 calories. Meanwhile, 1 gram of sugar has 4 calories. However, other research suggests that artificial sweeteners may have a negative impact on the food reward system and the gut microbiome, potentially leading to weight gain. When something with table sugar is consumed, the brain sends signals and dopamine throughout the body, creating a sense of fulfillment and satisfaction. However, when artificial sweeteners are consumed, the brain doesn't send those same signals, potentially leading to cravings and overeating, which can result in weight loss. Furthermore, the gut microbiome are the microorganisms that live in the digestive tract, and they are responsible for energy, thermoregulation, and appetite. And some studies have shown that artificial sweeteners may have a negative impact on this energy, thermoregulation, and appetite. And more about these topics can be read about in my literature review essay. And because of this contradicting research, I asked the question, how do artificial sweeteners impact the weight gain of mice? The hypothesis was that if mice are given artificial sweeteners, then they will have a higher increase in weight compared to the control and table sugar groups. The null hypothesis was that there will be no change in weight for each group, indicating that artificial sweeteners have no impact. The materials I used for this project were 5 cages, and each cage had a feeder, waterer, and wheel. And I also used 2 bags of 99% dust-free bedding, 12 pounds of a 22% protein, 5% fat complete mouse pellet, wooden sticks, cardboard tunnels, cut-resistant gloves, table sugar, aspartame, sucralose, stevia, an autoclave, a scale, graduated cylinders, a red, blue, and green marker, and then 15 female musculus mice. And working with mice does add another level of safety, not typical in a typical experiment. So to keep the safety standards high, I worked with Dr. Sarah Finney, who is a licensed veterinarian at Tri-County Animal Clinic. And then I also referenced the Jackson Laboratory Handbook on working with laboratory mice. And the species of mice used was the musculus because it is the species most genetically similar to a human, and it is the mouse that is most often used in lab experiments. And the Jackson Laboratory Handbook recommended that a food with at least a 16% protein, 4% fat content was used, and I used a 22% protein, 5% fat content, so that was met. And then I also utilized an autoclave, and I autoclaved all water that was given to the mice at 134 degrees Celsius over a 2-hour heating and cooling period to sterilize the water and kill any bacteria that may be in it. For the mice environment, I provided them with wooden sticks and tunnels to chew on because the teeth of mice are constantly growing. And I also provided them with wheels for exercise. And the Jackson Laboratory Handbook recommended that when handling the mice, you scoop them with your hands and you do not grab them by their tails because grabbing them by their tails has been shown to cause stress, and stress is not good for the animal or for the experiment. And female musculus mice were used because male musculus mice will fight, and that can cause stress, which isn't good for the animal or for the experiment. And for the procedure, I first divided the 15 mice into 5 cages, and as I added 3 mice into each cage, I labeled their tail with either a red, blue, or green marker to keep track of which mouse was which. And I also labeled the cages as either the control group, aspartame group, sucralose group, stevia group, or the table sugar group. And for the first 2 weeks, I just let the mice adjust to their new environment so they could get used to the light and day cycles, their water, their food, their new environment. And during these 2 weeks, I provided them with regular autoclaved water and 20 grams of food. And after the 2-week adjustment period, I got their initial starting weight. And based off of the total weight in each cage, I took the acceptable daily intake as calculated by the Food and Drug Administration for each artificial sweetener and table sugar, and I scaled down the acceptable daily intake for humans into the mice weight. And the acceptable daily intake for table sugar is 50 grams per 70 kilograms of body weight. For aspartame, it's 50 milligrams per kilogram of body weight. For sucralose, it's 5 milligrams per kilogram of body weight. And for stevia, it's 4 milligrams per kilogram of body weight. So taking the acceptable daily intake for the weight in each cage, I added that to 25 milliliters of autoclaved water, and I gave the mice a fresh 25 milliliters of water every day. And every week, I would weigh the mice and then recalculate the acceptable daily intake. So whether the mice lost or gained or their weight remained the same, they were always having the acceptable daily intake in their water. And I ran this experiment for 11 weeks, and then at the end of 11 weeks, I got their final weight. Throughout the experiment, I cleaned the mice cages once a week, and that's when I would replace their bedding and wipe anything that needed to be wiped down. And then every day I would spray around their cages with bleach spray just to keep the environment as clean as possible. And I also provided the mice with unlimited food so I could test the impact of artificial sweeteners or table sugars impact on the food reward system. And I could tell the mice had enough food because when I would go to clean their cage, I could see food crumbs scattered throughout. For the first week, the mice were content with 22 grams of food. And then the following week, I upped it to 24 grams. And then they had 26 grams of food given to them every day for the remaining of the experiment. And after 11 weeks, an ANOVA test was ran to study the effects of each treatment on the percent weight gain for each mouse. A p-value of 0.6861 was found, and an alpha value of 0.05 was used. So this fails to reject the null hypothesis, meaning that we conclude that there's no significant difference between the treatments on the mouse weight gain. Additionally, I ran an unequal variance test to confirm that the variances were equal, and I got no significant p-values, meaning the equal variance assumption holds, and I'm justified in using the ANOVA test. So to accompany these statistics, I made 2 graphs. The first graph compares the average weight per cage per week, so the sweetener versus the weight. And then the second graph compares the sweetener versus the percent body weight changed. And throughout the experiment, errors could have been made. The first issue I ran into was at my school. the scales only went to 3 decimal places in the grams. And because the acceptable daily intake for stevia and sucralose was so small, I found myself weighing such minute amounts of sugar, and so an error could have happened there. So to try and prevent this, I would batch prepare the stevia and sucralose for the whole week. So I would add 175 milliliters of autoclaved water and then add the total sweetener for the week to just try to get that number higher so the scale could actually recognize it. And I also couldn't control the amount of food and water each mouse consumed. So to try and combat this, I provided the mice with unlimited food because mice tend to have a runt and a leader, and I wanted to make sure that the runt could still get enough food. And then this also tested the food reward system. And furthermore, on week 8, one of the control mice died, and leading up to her death, she lost 20% of her body weight. And this could have thrown off the experiment. So to see if it did, I ran the ANOVA test with and without the control mouse, and whether she was included in it or not, there was no difference. The experiment was still statistically insignificant. An interesting find, on week 4 and 8, there was statistical significance. On week 4, the aspartame had a p-value of 0.0302, and table sugar had a p-value of 0.045. And on week 8, aspartame had a p-value of 0.0026. In the future, I could run shorter tests because of the statistical significance found at week 4 and 8 for aspartame and table sugar. And then I could also run longer tests because in the scientific world, 11 weeks is not a lot of time to run an experiment. I could also match the sugar consumption to the average American consumption because Americans consume more table sugar than artificial sweeteners because artificial sweeteners are hundreds of times sweeter than table sugar. And furthermore, I could test artificial sweeteners' impact on beverage consumption because throughout the experiment, I tracked the leftover water every day from each of the cages. And throughout the experiment, aspartame, table sugar, and sucralose consistently would drink most of their water, while the control and stevia groups would always have water left over. So in conclusion, artificial sweeteners do not impact the weight gain of mice because the statistical analysis ran was statistically insignificant. And here are some photos of the mice throughout the experiment. And then this is the weighing process for them. And then there are all 5 cages. All right, thank you.
Thank you. Any questions for Mackenzie? That was a great presentation. Thank you very much. Any questions? Right. Then thanks again. Now we have the third speaker. She couldn't be here today because of her schedule. Her name is Hira, and she's a senior at Bellbrook High School. She's interested in science, leadership, and arts. She has earned 2 state superior titles at the science fair. And outside of the academics, she's passionate about theater. And this is actually, she's at a theater play today. So I'm going to try sharing her video with you.
Hi, my name is Hira Nair, and this is my project, General Patterns for Onset of Symptoms in Retinitis Pigmentosa Patients.
Sorry.
RP typically starts with night blindness, which is a condition characterized by impaired vision in low light environments and gradually progress.
I'm just going.
Hi, my name is Hira Nair, and There, and this is my project, General Patterns for Onset of Symptoms in Retinitis Pigmentosa Patients. RP typically starts with night blindness, which is a condition characterized by impaired vision in low light environments and gradually progresses to visual field constriction, which is a progressive loss of peripheral vision resulting in a narrowed field of view, and visual acuity reduction, which is a decline in the sharpness and clarity of vision. The age at which onset of symptoms happens varies depending on the specific genetic mutation and other factors, leaving newly diagnosed RP patients uncertain about the nature of their RP progression. However, as we analyze current available research data on RP, a few general patterns begin to emerge. The objective of this research is to analyze these patterns and develop and test hypotheses related to onset of RP symptoms regardless of genetic mutation. Okay. Retinitis pigmentosa is an inherited group of retinal diseases that affect photoreceptor cells in the retina, specifically rods and cones. These cells are responsible for capturing light and enabling vision. In RP, mutations in certain genes lead to the progressive death of photoreceptors. This degeneration causes a decline in vision, starting with peripheral vision and progressing towards central vision loss. The cause of RP lies in genetic mutations that result in apoptosis or programmed cell death of photoreceptors. These mutations can also cause dysfunctions in ciliary transport, a process essential for photoreceptor survival. This degeneration can vary based on the type and location of mutations. RP follows three main inheritance patterns: autosomal dominant, which is the most common form of RP, responsible for about 30 to 40% of cases. A single copy of the mutated gene from one parent is sufficient to cause this disease. Number two is autosomal recessive, which accounts for 50 to 60% of RP cases. To develop RP in this case, an individual must inherit mutated genes from both parents. Finally, we have X-linked RP, which is responsible for approximately 5 to 15% of RP cases. And this form is associated with mutations on the X chromosome and generally affects males more severely. Understanding these patterns is very crucial for genetic counseling in determining the expected course of disease. RP can also be classified into two broad categories: non-syndromic RP, which is the most common form, making up about 80% of cases. It involves only retinal degeneration and no other systemic conditions. The symptoms typically start with night blindness and progress to visual field constriction and visual acuity reduction. The other type is syndromic RP, which is associated with other systemic conditions. A well-known example is Usher syndrome, which combines RP with hearing loss. Although it is less common, it is still a very important part of the RP spectrum. However, my study focuses on the more prevalent non-syndromic form of RP. The central question of my research is, can we identify general patterns for the onset of night blindness, visual field constriction, and visual acuity reduction in non-syndromic RP patients? To answer this, I compare a synthetic reference sample, which is based on published research and published literature available on RP, with the survey sample collected directly from RP patients. By doing so, I aim to determine if symptom onset patterns in real-world patients match the predictions derived from genetic data and epidemiological studies. To formulate my hypothesis, I first collected data on the genetic mutations commonly associated with RP. The pie charts on this slide illustrate how these mutations are distributed across the RP population. These charts represent the prevalence of mutations within each inheritance pattern: autosomal dominant RP, autosomal recessive, and X-linked, showing the proportions of each mutation in the broader population. The visual distribution from the pie charts was essential because it guided the way I assigned weights to each genetic mutation. Using this data, I analyzed peer-reviewed studies and genomic databases related to non-syndromic RP. This review helped me identify common genetic mutations, prevalence rates, inheritance patterns, and the typical progression of symptoms. I used this information to generate a synthetic 1,000-person reference sample. This reference sample served as a baseline based on genetic and epidemiological data to estimate how symptoms typically progressed in non-syndromic RP cases. Once I had a clear picture of the distribution, I used this data to generate the 1,000-person reference sample. The sample was created by adjusting the prevalence data for each mutation. Genes with a known prevalence for mutations where prevalence percentages were available, I weight adjusted them to reflect their proportion in the overall RP population. For example, if an autosomal recessive RP mutation had a 2.5 prevalence, I assigned it with a corresponding weight of approximately 14 in the reference sample. For mutations with unknown prevalence, I distributed them uniformly within the inheritance patterns unknown category. If 50% of autosomal dominant RP mutations had unknown prevalence, I adjusted the weight of these mutations accordingly to match the overall RP prevalence. This method allowed me to create a reference sample that accurately mirrored real-world RP prevalence. The genetic profiles of each individual in the sample were reflective of the actual distribution of RP mutations, and this formed the foundation for estimating symptom onset across the RP population. For the reference sample analysis, I first calculated the weighted means and standard deviations for the onset of the three key symptoms: night blindness, visual field constriction, and visual acuity reduction. This allowed me to get a sense of the central tendency and variability of the symptom onset times across the 1,000 simulated cases. To further evaluate the distribution, I generated bar plots and probability density functions, which helped me assess whether the onset data followed a normal distribution. From these distributions, I then identified the 70th, 80th, and 90th percentile onset timelines for each symptom, which served as the key reference points for comparison with the survey sample. After analyzing the reference sample, I found that the onset of these symptoms follows a normal distribution. In this slide, you can see the bar plots that illustrate these distributions. From the data, we can clearly identify the 80th percentile thresholds for each symptom. For night blindness, the 80th percentile threshold falls around age 30, which is in decade 3. For visual field constriction, the 80th percentile threshold is near age 40, which is in decade 4. And then for visual acuity reduction, the threshold is around age 50, decade 5. These values are critical because they set the baseline for when we expect these symptoms to manifest in the general RP population. So, based on our analysis of this publicly available research, the study hypothesizes that 1, 80% of or more of RP patients who exhibit night blindness will have the symptom by decade 3 or age 30. 2, 80% or more of RP patients who exhibit visual field constriction will have the symptom onset by decade 4 or age 40. 3, 80% or more of RP patients who exhibit visual acuity reduction will have the symptom onset by decade 5 or age 50. These are predictions based on genetic data and symptom progression trends. To validate my hypothesis, I decided to compare two data sets: the reference sample that I had created using my research and a survey sample to validate whether the symptom onset patterns seen in the literature aligned with real-world patient experiences. Once I had generated the reference sample, which served as the baseline for symptom onset predictions, I moved on to analyzing the real-world symptom onset patterns of non-syndromic RP. To do this, I collected data from a group of 164 individuals diagnosed with RP. However, since I was focusing specifically on non-syndromic RP, I had to filter out 27 responses from individuals with syndromic conditions, such as Usher syndrome, leaving me with 137 non-syndromic RP cases for analysis. I made sure that the survey sample reflected a diverse range of individuals, taking into account variations in age, gender, and disease progression. This approach was essential for ensuring that the data was representative of the broader non-syndromic RP population, which helped improve the accuracy and generalizability of my findings. The survey asked participants to recall the onset of three key symptoms of RP: night blindness, visual field constriction, and visual acuity reduction. For each symptom, the participants provided the decade at which they first noticed it and described the severity of it at that time. With this data in hand, I was able to compare the self-reported onset times with the expected symptom onset patterns derived from this reference sample. This comparison not only validated the trends seen in the published literature, but it also allowed me to assess how well these trends aligned with the lived experiences of RP patients. Next, for the survey sample analysis, I used bootstrap resampling. I created 1,000 resampled datasets from the survey sample, each containing 1,000 randomly selected responses from the original RP responses. For each resampled dataset, I calculated the frequency of symptom onset by the 80th percentile age as established by the reference sample. This method allowed me to test how closely the real-world data from the survey sample aligns with the expected symptom onset trends. The bootstraps process helped ensure that any conclusions drawn about the alignment between the two databases were statistically valid and reliable. Analyzing the survey sample plots, we can clearly infer the strong agreement that the sample has with our hypotheses. The probability of RP patients reporting night blindness by the third decade had a 95% confidence interval, fluctuating between 83% and 88%, with a mean of 85%. Patients reporting visual field constriction by decade 4 had their 95% confidence interval fluctuating between 86% and 90%, with a mean of 88%. Finally, patients reporting visual acuity reduction by their fifth decade fluctuated between 87% and 91% for a 95% confidence interval, with the mean centered at 89%. These findings confirm that the symptom onset patterns from the survey sample align closely with the expected trends from the reference sample. This validates the reliability of the hypothesis, reinforcing that the predicted symptom progression is consistent with real-world experiences in non-syndromic RP patients. Analysis of the samples from survey responses demonstrated a consistent pattern for symptom onset reporting by RP, diagnosed patients with 80% or more reporting night blindness by age 30, visual field constriction by age 40, and visual acuity reduction by age 50, validating our hypotheses about onset of night blindness, visual field constriction, and visual acuity reduction symptoms in RP patients. In conclusion, based on the comparative analysis of the survey responses and publicly available research, I can confidently accept our null hypothesis. I conclude that RP patients will report night blindness by age 30, visual field constriction by age 40, and visual acuity reduction by age 50. Research further suggests that while some RP patients may experience severe manifestations of visual field constriction and visual acuity reduction qualifying for legal blindness in later decades, the complete loss of vision is relatively rare. Most patients retain some degree of central vision throughout their lives. This study provides valuable insights into the typical progression of RP, offering clarity to patients. It could help them plan their lives and develop early intervention strategies. Future studies incorporating a broader range of genetic mutations and larger sample sizes will help refine these findings and provide more detailed gene-specific patterns of RP progression. Thank you so much.
Yes, so